Ok, pochi secondi.
Grazie.
Okay, we are on air, we can start.
Okay, thank you very much, Manolo. So good morning, everybody, and welcome to the last webinar organized for the SIICA School of Immunology 2020. So it's a great pleasure for me to introduce Professor De Gregorio. Professor De Gregorio, he's the head of the Research and Development Center at GSK Vaccine here in Italy. He is responsible for the discovery and development of several vaccine targeting bacterial infection, such as, for example, bacterial meningitis and other infection caused by antimicrobial resistant pathogens. And he is also responsible for the discovery and development of novel vaccine adjuvants, as well as a novel breakthrough vaccine platform. So thank you so much for accepting our invitation, Professor De Gregorio. So please, thank you.
Thanks for the introduction. So first, I'm not professor. My career is, I have a scientific background, of course. I've done few words about myself before I started. I've done the university degree in biology with Irene Bozzoni in Rome. Then I did the PhD at the EMBL on, and all the university and PhD was about RNA. And I'm saying that because during this presentation, you will see that the RNA cross vaccines a lot, especially in the COVID crisis. And I've done a lot of RNA control, processing, and translation in my previous career. Then I moved to innate immunity, and I did a postdoc in the lab that essentially discovered toll-like receptors. It's the lab of Bruno Lemaitre in France, where I spent like 3 or 4 years. And I worked on innate immunity in Drosophila, discovering new innate immune factors. And then from that, I thought, okay, now what I can do that can have better impact than just publishing papers. And I moved to the industry. And because I was working in innate immunity, the straightforward jump was from Drosophila innate immunity to vaccine adjuvants, because as you know, vaccine adjuvants are, as you probably know, are in general the agonist of innate immune receptor like pattern recognition receptor. So it was a very fascinating story. We developed new adjuvants. At that time was just the beginning of this era in innate immunity, and I worked a lot on vaccine adjuvants. And then I made my career in the company, and I don't give you all the details, but now I'm the head of research and development. So not only discovery, but also development of vaccines in Siena. We are Center of Excellence for Bacterial Vaccines. We have several programs for global health and also for vaccines with commercial value. We do not work on viral vaccines at the moment, although we have worked in the past on viral vaccines, in particular the pandemic vaccines adjuvanted with MF59, where we were Novartis. So I have experience, and we have a lot of experience on adjuvants for viral vaccines. Normally, I focus more on bacterial vaccines in my presentations. This time, because of COVID, I have readapted the presentation to focus more on the novel technologies for human vaccines, which are indeed relevant for viral vaccines, and in particular to address one big question, that is, how can we rapidly respond to emerging pathogens? As you see, as you know, if there is an emerging infection, vaccines are always late. They have always been late. Now, sometimes they are people start to work and then the infection goes away and the vaccine is never developed. Sometimes gets developed, but years after the emergency where it's not needed anymore. What we're trying to do this time is to have a vaccine in record timelines to be available when it's needed. So I would say maximum next year. And that's the introduction. Let's go to the presentation. So I like to start from the beginning. Vaccines, you probably know that, so I apologize for people that already know that, but it's important to know that the conventional vaccine approach, you know what it is based on Pasteur principle. You have to isolate the pathogen, inactivate, and inject. And this has been the way vaccine has been done for 50 years. Isolate the pathogens, then you need to grow the microorganism And then you can do different things that are done for most of the vaccines that are available. You can inactivate microorganisms, and so you can have all-kill vaccines, for instance, rabies vaccines, typhoid vaccines, inactivated polio vaccines are made this way. You can attenuate the microorganism and so have a live vaccines, this is live attenuated vaccines, rubella, mumps, measles, varicella, oral polio are made this way, that they're still used and they're very effective. Or you can chemically detoxify toxins that are present in the microorganism, and tetanus and diphtheria have done this way. You can isolate the polysaccharide, and there are still some pure polysaccharide vaccine for pneumonia and meningitis. Or you can grow the microorganisms, in this case in eggs, purify the protein, and then you have subunit vaccines like influenza and pertussis. Those are the Pasteur vaccines, and these vaccines have been extremely effective to almost eradicate infection. For polio is almost an eradication. Smallpox is not here, has been eradicated through a classical Pasteur live attenuated vaccines. Pertussis is down 94%, diphtheria 93%, tetanus 89%, you can see them. So the vaccines made with Pasteur principle have been extremely successful, but for some microorganisms, you cannot use this method. And other methods have been utilized. So conventional Pasteur approaches in this slide on the top. Some new approaches that have been used are glycoconjugates. Why? Because polysaccharide don't have a T helper response. So essentially, you don't have a good T cell support to the immune response. Therefore, you don't have memory, you don't have a good, strong isotype switch, you don't have strong avidity. Generally, the polysaccharide vaccine lasts for 1 year, and then if you boost, you reduce rather than increase the antibody response. If you conjugate the polysaccharides to a carrier protein, then you provide the T cell help from the carrier to the polysaccharide, and then you have strong isotype switch, robust memory, and therefore glycoconjugate vaccine has been a big improvement in vaccine that were not very effective for polysaccharide. Some pathogens cannot grow, like HBV, you cannot grow the virus, and therefore recombinant proteins have been used to make the vaccine. Recombinant proteins, you will see, most of the time, they need an adjuvant to elicit a strong response. Recombinant viral-like particles are very similar to recombinant protein. The only difference is that they assemble in particles, and they also made it adjuvant. And then there are already vaccines that have been licensed that use viral vectors, replicative or non-replicative vector, as you will see. Those are the things that are licensed. All the rest you are hearing about these days, mRNA, self-amplifying RNA, DNA, those things have not been licensed yet. There is no proof of concept that works. These things that you see here, they already have vaccines on the market. I just move. So for the immune response, this is for immunology. I think this slide is important. Which type of response you get with this type of vaccine? So live attenuated vaccines, but also viral vectors, and also DNA and RNA vaccines. They elicit a complete immune response. They have already an adjuvant built-in activities, because either they make an infection or they are itself, by themselves, DNA with CpG, RNA, because just as an RNA and they elicit a lot of RNA sensor, they activate dendritic cells, so they don't need an adjuvant in general. There's no viral vector DNA or RNA vaccines that has been used with an adjuvant. They really don't need an adjuvant. They are made, the protein is made in the cytoplasm and is presented by MHC class 1. So they induce normally a very good CD8 response, but they can also be presented to MHC class 2. They induce also a very good CD4 response. And they have a very good also antibody response, because follicular helper T cells are generated, and also the antigen is made and binds and cross-link the B cell receptor. So you get a very good antibody response. So with this vaccine in red, I don't know if I can write here, but anyway, with this vaccine in red, live attenuated, others, you get good effector memory CD8, good helper and effector CD4, high affinity antibody, memory B, and CD4 memory. That's what you get. kill all organisms. They don't need an adjuvant because when you make the vaccine, there are RNA, DNA, pumps are present. So generally, they don't need an adjuvant to elicit a robust immune response. They are well presented in MHC class 2. They have a very good CD4 and antibody response, but they do not induce CD8 response. Same with protein vaccines. Protein vaccines are so based on protein, viral-like particles, and also glycoconjugates. In general, they made it adjuvant to work and induce a good CD4 and B-cell response, but there is no evidence for CD8 response in response to this protein-based vaccine. There is evidence that with adjuvants, you can have cross-presentation and have CD8 response in mice, but when we move to primates and humans, we have never seen an adjuvant that, although the CD8 was measured in mouse system, was not measured in non-human primates and human beings. So essentially, we don't have at the moment an adjuvant that is able to induce a CD8 response for protein vaccines. As I said before, polysaccharide vaccines, they're very good in cross-linking. The IgM and IgG were present on B cells, but they only give low affinity antibody and no memory. Those are the classes of vaccines that are present, and this is the immune response that you can elicit. I have to say here as a comment, all vaccines that we have licensed are based, when there is a correlative protection, is based on an antibody response. There are still questions whether the cellular response to vaccination really induce a protective, contributes to protection induced from the vaccine. Maybe I can stop here for a while if there are questions on this general immunological part. I mean, you know about immunology probably more than me, but if there are questions, I can take a question here.
So I invite students if they have questions on this part.
Otherwise, move on.
Apparently no questions so far. Thank you.
It's too basic, probably. Okay, so those are the vaccines that I mentioned. And you can see for each class of vaccines, where are the licensed vaccines. So there are many vaccines. They prevent 25 million deaths every year. And those are I don't want to go through the entire list, but you can see that for each class of vaccine, there are several examples of vaccines that are working and are making their job. I mean, I don't want to go into the details here, but if you're curious to see what are the pathogens that are prevented by vaccine and from which vaccine, you can keep this slide as pre-reads. We don't have to spend words here. There are many vaccines, though, that cannot be made for easily with the class of, with the technologies that we are working about. You can see, this is a review that I wrote with Rino Rappoli in 2014. And this is the list of very important candidates that are not preventable with vaccination at 2014. You see bacteria, of course, tuberculosis, group A, group B, streptococcus, staphylococcus aureus, Shigella. I mean, you name it. There is really many, and some of them are really bad and major killers, in particular tuberculosis. Viruses, some key viruses like hepatitis C, respiratory syncytial virus, CMV, there's no vaccines for these things. Despite these viruses have been there for forever, for 100 years, and people have tried to defeat these pathogens with vaccination. Nobody succeeded. And I'm saying that because when people say, okay, COVID, you're going to have the vaccine in 1 year. Well, consider that RSV, that is another RNA vaccine, has been there for forever. And since vaccination exists, we have not been able so far to make an RSV vaccine. So I don't want to depress you, but I mean, guys, it's difficult. And it's not obvious that if there is a pathogen and you can isolate and you can sequence the pathogens the day after you have a vaccine. The parasites, also there are many. Plasmodium for malaria is the key one. And there is not a single therapeutic vaccine that works. So we don't have a vaccine to cure existing chronic infections or other diseases. So there is a big war of things that needs to be prevented by vaccination. And the asterisk are the vaccines that have been licensed since 2014. So there is one malaria from GSK that now is in a pilot in Africa and gives a partial protection for malaria. It's not the ideal vaccines, but this is a vaccine that can make a difference. There is a vaccine for dengue made by Sanofi and is only I mean, I don't want to cut this. I mean, it's a viral vector, chimeric vector. It has been licensed, and this is an Ebola vaccine. There's also a replicated vector that has been licensed. So for most of the pathogens, we still don't have a vaccine. So new vaccine technologies are required for these difficult targets. That's clear, even beyond the one that I just mentioned in the previous slide. That's the first reason why we need new technology. Second reason, antimicrobial resistance, huge problem. 2000 to 2010, antibiotic consumption rose by 36%. So we use more and more antibiotics. If you look at 2010 to 2020, I'm sure when the report will be issued, we will see even more increase of antibiotics. It's antibiotics for human use that is appropriate because you have a bacterial infection, but can also be antibiotics for use that is not appropriate because you get antibiotics even if you have a viral infection because it's just in case, right? So there is still an abuse of antibiotics for human. Is antibiotics used in agriculture for crops and for animals that can also have an important implication for antimicrobial resistance generation for bacteria that cross species from animals to humans. So antimicrobial resistance as a result of that has increased and is increasing. There are around 700,000 deaths annually, but in a few years, people are talking about, I didn't put here that, but in 2035, I believe was the year, it has been forecast, the forecast has been 10 million deaths. Sorry, by 2050, right? So in 2050, we may have 10 million deaths for MR if we don't do anything. It's bigger than COVID, it's huge, and it's just there. We see these things progressing. We see outbreaks of Klebsiella, carbapenem resistant. We see outbreak of Staphylococcus meticillin resistant. We did outbreak of gonococcus that cannot be cured with any antibiotic already. So there are some bacteria that literally cannot be cured, and they're there. So we need to do something to prevent antimicrobial resistance, and we think that vaccines is the best way to do it. Why? Because vaccine can directly prevent infections caused by MR, but the use of vaccines can also reduce the infection rate for bacterial and virus, limiting proper and improper use of antibiotics. The most important thing to say is that while for antibiotics you get resistance, and you get resistance all the time. There is not a single antibiotics that did not induce resistance few years after implementation. The polyclonal nature of the response to vaccines does not generate resistance. The epitopes that are targeted by the vaccine are always multiple epitopes, and the bacteria are not able to evade the response made by vaccination. Therefore, you don't have antimicrobial resistance. Once you have the vaccine, as you've seen from the previous slide with tetanus, diphtheria, the infection is almost eradicated, and sometimes you get a 1%, and sometimes you don't see it anymore. So this is really the long-term solution for antimicrobial resistance. And in my R&D center, we are focusing, we have a lot of targets in discovery to address that. It's not the hot topic today because of COVID, but what we can learn from COVID is that we have to anticipate. We cannot wait for the next pandemic to come, because if we wait, the vaccine will be late. And so for MR is the same. We cannot wait the big antimicrobial resistance outbreak to come. We have to anticipate and have a vaccine that is ready before. That's why we are working a lot to anticipate that for most of these pathogens. Then the next reason why we need new technology is speed. And it's what I've been saying. Vaccine research and development is super complex. I wrote here, it takes several years, 7 to 10, for discovery to vaccination. Depends on the pathogen, but generally we spend 2 to 3 years in discovery to select the best candidate. Depends on the pathogen. This can be shorter, but it takes some time because you want to go in human with the vaccine that are the highest probability of success. And there are always several options that you can, of antigens, selection, as you will see, not only the selection of the antigen, the identification of the antigen, but the conformation, the structure of vaccinology. There is work to do to improve the probability of success. This is not what we're doing with COVID, where we went straight with the first target we could think about. Then you have to do clinical trial material, and you will see what is the critical point here. But with the classical platform, it takes years, 1 year at least, to have GMP material for a clinical trial. Then you have to do a phase 1 for safety. Then you have to do a phase 2 for safety, immunogenicity, and selected dose. And then you have to do a phase 3 for efficacy. And all of these steps are highly regulated. And so you end up with generally 10 years, more than 10, for vaccine discovery. If you have an outbreak like COVID, tell me who is interested in a vaccine in 10 years from now. So we need new platform to accelerate the response to vaccination, and for vaccine discovery, vaccine development, but also for vaccine scale up and production. Getting a manufacturing site for a vaccine requires many, many years. So, and this slide probably is the one that will capture more the attention from you, because it's really the question that we are trying to address for COVID. How can we speed up discovery? How can we speed up development? How can we make, in this case, billions of doses of vaccines ready for people to contrast the epidemic? And you will see in this presentation, what I will try to do is to show what are the different technologies that are promising, and most of them are used for COVID, and how do they address the problem of speed and in this situation. So should I move on with the different sections and one by one, or you have some questions on this part? The
So students, if they have
give me...
Yes.
It seems like we have no questions.
Yes, there are no questions apparently. No.
Good. So I move on, and so let's start with antigen identification and structural vaccinology. This is key in general, also for COVID. So how do we select the antigen for vaccines? The place where I live, where I work in Siena, is the place where the reverse vaccinology approach was first identified for MenB by the team of Rino Rappoli. It's interesting, it's not key for viruses, but I think it's still interesting. For the first time, for meningitis B vaccines, in order to identify what is the target, the antigen to use, it was used this genomic approach. It was for the first time, it was a long time ago, meningitis B was sequenced, prediction of protein expressed on the surface of the bacteria was made. 350 proteins were successfully expressed in E. coli, immunized mice, and sorry, I have to close the door. We have selected the candidate antigens for their ability to induce essentially antibodies in preclinical model that are able to kill the bacteria in a serum bactericidal assay, which is the correlate of protection for the vaccines against meningitis. So with this method, we have essentially developed the meningitis B vaccine, Bexsero, that is made of 3 proteins and an IMV. So this is one way to identify antigens. What we're doing more and more now is, instead of testing all these proteins, is try to find shortcuts. And how can you find a shortcut to identify the antigens for both bacteria and virus? Is to interrogate humans, not do preclinical assay, but interrogate humans, thanks to the huge advancement of B-cell technology. So I thought to put the next slide, because for an immunology course, I think this is extremely important. So now what can we do? We can go to people that have been vaccinated or infected. In case of if I have to find an antigen for meningitis B now, I will go to a human being that survived meningitis B infection if I don't have the vaccine. You can collect B cells, and you can, what you can do now is either take plasma cells or memory cells and screen each individual B cells for the ability to induce antibodies that are functional, that kill the bacteria or neutralize the virus. And after isolating the antibody, by looking at the sequence of the antibody, it is possible to identify, express the antibody, those identify the antigen and the epitope that is required for the protection functional assay that you're doing. This approach has been extremely useful in viruses to identify HIV broadly neutralizing antibody, and therefore epitope that can generate broadly neutralizing, for CMV, for RSV, and universal flu, as you will see. So this slide is show you what can be done. Antibody repertoire can be expressed, chronotypes can be expressed, or B cell can be immortalized with EBV, and you can directly screen with functional assay on all the B cells that are immortalized. Or if you know when the infection occurred, you can get the plasma cells 1 week after infection or after vaccination. Most of them would be responding to the pathogen or to the vaccine, and so you can interrogate plasma cells. So those are the 3 ways people have used. Then you can run a functional assay, identify antibody that are functional, then use biochemistry to identify antibody targets, and then use this epitope information to identify the antigen that expose the epitope, or you can immunofocus on the epitope. You will see there are different ways, but essentially design your vaccine antigen based on the knowledge that derives from functional antibodies. This is where the world is going. This is the direction that the world is taking for vaccines that have not been possible for years, like HIV, CMV, and RSV, at least. We could not do that for COVID, but I have to say that the work has been done for SARS. Actually, the first paper showing a B cell work to isolate a neutralizing antibody from Antonio Lanzavecchia comes actually from SARS in 2003, I believe. And so we have knowledge now on the spike of SARS that derives from the B cell analysis of neutralizing antibody. And this knowledge was used as soon as the sequence of COVID-19 came out to try to make a shortcut and try to design an antigen that exposes the neutralizing epitope that have been identified for SARS. I can tell you there are a lot of labs that they're doing that, exactly that now for COVID, essentially interrogate the B cell repertoire of COVID people that survivors, to identify potent neutralizing antibody that can be used in therapeutics, but also to guide the future of what can I say, COVID cross-neutralizing vaccines, pan-coronavirus vaccine. This is probably the most exciting things that is happening in research, because could give a solution very soon, essentially a neutralizing antibody that can be given to people that are very critical, in a very critical medical conditions, are going to intensive care, and there are already candidates, but can also provide a solution for the future for a pan-coronavirus vaccine. Examples of this approach are, the most notable is probably influenza. You know, there are several clades of influenza, H5, H2, H5, H7 are the most well-known pandemic avian flu influenza, while H1 and H3 and B is not here because there is only influenza are the ones that are present in the seasonal vaccine. So the objective of a big medical need would be to address influenza as a universal vaccine. So you don't have to change every year, but will address the influenza for multiple years and could also protect for pandemic in case pandemic will occur. So this search for an antibody and eventually vaccine that cross-protect all influenza was considered no way in many years ago, until many labs have started to interrogate B cells for flu, and they found antibodies that are able to neutralize different clades of influenza, even the group 1 or the group 2. But here in 2011, the team of Davide Corti and Antonio Lanzavecchia and Bellinson, they found a very interesting antibody that is able to neutralize all of them. What they did was to collect blood 1 week after an influenza vaccine shot. If you do that, you have plasma cells in the blood that are 60–70% of them are directed against the antigen that has been used for immunization. So in this case, hemagglutinin. They isolate plasma cells, they culture them in single cells with the interleukin-6, they screen the supernatant for neutralization, and they have identified the antibody that could neutralize the influenza. They identified this F16 antibody that is able to neutralize both group 1 and group 2 influenza HA. There are other antibodies that have been found, but this is pretty remarkable. And most of these antibodies recognize the stem of the HA, so not the immunodominant receptor binding domain in the head, but the less dominant stem. So essentially, you can find things that are not obvious at all, that are part of the protein that are normally not considered for vaccines, but now people start to consider because they could be the epitopes that generate a broadly neutralizing vaccine. So the problem is, once you know what is the epitope that generates special antibodies, very efficient antibodies, in this case, special because they cross-neutralize, how do you elicit from a vaccination a response to these vaccines? If you use the HA as it is, well, this is not the dominant response that you get. The dominant response is for the head. So you need to design a vaccine to focus on this epitope and allow for for this antibody to occur. And so this paper is from 2011. In 9 years, there are several approaches to induce antibody against them. I don't have a slide that summarizes all of them. Those are 2 examples. Some people have put the stem without head to avoid the immunodominance of the head and put them in a nanoparticle to have stems that are able to cross-link the B-cell receptor and also induce antibody. Others have just used a trimeric stable stem. I can tell you that other teams, and we have been working with this team, has used a chimeric approach using a first immunization with the head from avian flu and the stem from one clade, and then immunize with another, boost with another protein that has a different head, and therefore in the boost focus on the response to the stem that is doing the boost. There are several approaches, but the race is there. So the point I wanted to make, through monoclonal antibody, there was a proof of principle that is this possible to cross-neutralize different influenza. It is possible to have a universal influenza vaccines. And now what people are doing is work using this epitope knowledge to design a vaccine that can induce this cross-neutralizing vaccine. It's fascinating. You can imagine the same is going on, and of course it is going on for HIV, for all the pathogens where cross-neutralization, breadth of response is key. 2 other examples, and then probably have to stop because I'm talking too much, of the use of monoclonal. In the case of RSV, there are, this is very important if you consider COVID. So RSV is a similar virus, it's an RNA virus, as a sort of spike, that is the RSVF, that is the one that binds receptor and allows the virus to enter into the cells. RSVF has 2 conformations, one that is pre-fusion and one that is post-fusion. The post-fusion conformation for viruses is generally the more stable. So when you make it in CHO cells, or you want to make a recombinant protein, it tends to adopt the post-fusion conformation. And we have been working with post-fusion conformation antigens for years because nobody knew even what was the conformation of the pre-fusion F. And essentially, a lot of vaccines failed, in my opinion, or were not enough protective because we were using vaccines that had the majority of the F in the post-fusion conformation. Of course, the post-fusion conformation is the one that is created inside the cells after infection. It's not the one that you need to neutralize. By chance, F post-fusion has one epitope that is the same of the pre-fusion, even if it changes completely conformation. And so there is a partial effect, but it's not enough. So if you want a vaccines against F, you need to have an antigen that is in the pre-fusion conformation, the conformation of the virus before binding the receptor. As I say, nobody knew what was the conformation until in 2010 to 2013. A lot of work has been done to identify monoclonal antibody, in this case D25, that binds only the pre-fusion conformation. By using this antibody, it was possible to have a crystal structure for the first time of the F pre-fusion conformation. And thanks to the work, to the structure information, what has been done by McLean and the team of McLean and NIH was to stabilize the prefusion by inserting some disulfide bonds and also by using a folder that you can see here in the bottom of the slide. So essentially, thanks to the antibody, we were able to identify the structure of the prefusion. Thanks to the structure of the prefusion, we were able to stabilize it and design an antigen that doesn't revert to the post-fusion conformation and that we can now use for an effective RSV vaccine. COVID is also in a pre or post-fusion conformation. The antigen that are being used now have been modeled to be locked into the pre-fusion conformation through a similar strategy. So thanks to the science on RSVF, I would say it was possible to accelerate also the candidate for COVID by making some assumptions here, but that I think is probably going to work. So having a stable pre-fusion state for RSVF and for COVID-19 is a key attribute to get for the antigen. Another example is CMV pentamer. CMV is another pathogen that has been there forever. It's a huge medical need, especially for congenital infections. You can get congenital infection for CMV in the first trimester of pregnancy, and this leads to big, very important effects in the newborn. It's a huge medical need, especially for seronegative women. So it has been there, but how do you make an efficient vaccines? Again, here, through the identification of a very potent neutralizing antibody, 916-FAB, you see here, he was able to pull down a structure that was not known, this CMV pentamer. So essentially, what we realize is that there is a pentamer made of 5 CMV protein that is key for entering the cell, especially epithelial cells. And if you neutralize this pentamer, you have a chance to have a very efficacious vaccine. We still need proof of concept, but essentially the antigen, the pentamer, was identified thanks to the identification of the antibody, selected by functions, and then through the biochemical pull down. So this shows that you can use monoclonal antibody to identify protective epitope in flu, or to design the right antigens in the prefusion state, like RSVF, or to identify an antigen that was not known, like the CMV pentameric complex. This is I think, is 3 key examples of how important it is to use human immunology to design innovative vaccines. And okay, maybe I pause for a second here. If you have questions on this part or on any slide that I presented before.
So students, do you have questions so far? I have a short question, let's say comment. I would like to know which is your opinion about machine learning and the speed up of epitope prediction, let's say. So do you think that artificial intelligence can help us in identifying epitopes better than antibodies?
Well, I don't know. We are trying to do that. We have a collaboration in US with the Livermore lab where they're doing artificial intelligence. I think that artificial intelligence can help along the monoclonal antibody information. I don't think that the 2 things are alternative. So essentially, if you have an antibody that is telling you what is the epitope of the protein that you have to target, I think artificial intelligence can help you to generate the right immunogen. So to select out of the many structures you can have, what is the best structure to expose this epitope. For epitope identification, I'm not sure. I think that the monoclonal are more promising. I mean, there is a proof of concept. But I think that artificial intelligence, indeed, I mean, what has been done for the work of Bill Schiff is extremely important, because he started with the HIV neutralizing antibody, and essentially what he's doing through artificial intelligence and machine learning is trying to find out what is the antigen that I can use to elicit this cross-neutralizing antibody. The interesting thing is that these antibodies are not generated by they are essentially generated both by the germline, right? So you need an antigen that induce and start from the germline repertoire. And so there is an entire machine learning exercise to identify an antigen that can induce the germline that is totally designed by the computer. So that's one example where they push really, really far. And so what they're doing is to have an evolution of antigens. The first that act on the germline, the second already on some antibody that are the somatic permutation, and then following until you can generate the cross-neutralizing antibody. So there is an exercise that where you need the computer to assist. But yes, I believe that machine learning plus real information from the antibody together can really boost the acceleration of non-natural antigen configuration, not the one that is on the virus, but something else that can do better than especially if you want to have vaccines that do better than natural infection, like in the case of HIV, for example, or HCV. Yeah.
Thank you.
I have also a quick question. So how frequent can be the possibility of serotype replacement in case of vaccine that against antimicrobial resistant pathogens?
Well, for serotype replacement has been demonstrated for pneumonia, for pneumo vaccine. So you know the story. There was a 7-valent pneumo vaccine. And when they introduced the 7-valent, is an AMR, by the way. So pneumonia is one of the pathogens that get antimicrobial resistance. When they introduced the 7-valent vaccines, it was very effective against the 7 polysaccharides that were conjugated to cream, in this case for pneumo. But we started to see other serotype that not only were not targeted by vaccination, and we knew about that, but started to increase in terms of infection and pneumonia. So in the case of pneumo, by preventing the infection of some serotype, you increase the niche for other serotypes, and there was indeed in strain replacement. In fact, now there is a pneumo 15-valent in phase 3, and there is a pneumo 20-valent in phase 2, and in this case there will be a continuous catch-up to get the vaccines that also address the new serotypes where there is strain replacement. So it is possible. So far, we've seen that only for pneumo. I have to say pneumo has around 90 different strains circulating. So it's a bacteria where this can happen. For other bacteria, we haven't seen it. We haven't seen it for meningitis, where there is an ACWI. We haven't seen it for Haemophilus influenza, as far as I know. So for other bacteria that also, at least for which we use the capsule or the polysaccharide, the standard polysaccharide, similarly to pneumo, we haven't seen it. It is something to monitor. What I say is that you don't have resistance to the vaccination on the pathogen that you target, so essentially the same serotype. Indeed, you can have a strain replacement and have another bacteria, but it's another bacteria species. Sorry, it's the same bacteria species, but it's another bacteria serotype that can diffuse more. This is something that is true and has to be monitored. For instance, for Klebsiella, it may be a similar situation. There are several serotypes. People are starting to work on Klebsiella because of the resistance to carbapenem, and we had an outbreak in Tuscany last year, which was really bad, right? So it's one of the big AMR pathogens. If you want to address the thing by like pneumo, by addressing the key, by using as an antigen the polysaccharide, and by addressing the key serotypes based on polysaccharide, I think there is a risk that you have serotype replacement has to be monitored. For protein-based vaccines, we haven't seen. So for toxin-based vaccines, we haven't seen the not strain replacement, but evasion of the immune response. For protein-based vaccine in general, we haven't seen it. The only case is pneumo so far, but it's a point of attention that has to be, and there is a response. You can extend the valency of the vaccine to the other serotypes in case it happens.
And I have another. Sorry, another. I mean, is more just a curiosity. I mean, are there available studies which are defining, I mean, the impact of this kind of vaccination on the microbiome composition in patients? I mean, this vaccination, I can imagine, may have a huge, I mean, impact on the microbiome composition, so of the people. I don't know if there are studies or... Yes,
there will be more and more. It depends which pathogen you are targeting, because of course that there are, if you have a vaccine, okay, so let me start like that. Most of the vaccine that we did are for pathogens that are only acquired during infection, and therefore you see RSV when you get RSV infection. It's not part of the commensal flora. But for Klebsiella, for E. coli, even expect for other for Staphylococcus, now we are more and more targeting, especially bacteria that are present in the population and become a problem only during surgery or under special conditions. For these vaccines, and in AMR we have many, I think it will be important to monitor the impact on the microbiota. So for the vaccines that are out there, there are some data, but most of them are not really targeting a commensal. But for the new generation vaccines, it is actually something that will be definitely done. We have a vaccine for COPD that target non-typable and Haemophilus and Moraxella catarrhalis. Non-typable is present in the lung of many people in COPD, and we will address what is the impact on other commensals and on other bacteria there. So it's a question for the future more than a question for the vaccines that we already have. But it's an important question because indeed you may have a perturbation on the microbiome with some side effect due to that.
Exactly,
I mean,
exactly.
We cannot exclude that. We cannot exclude that.
Thank you.
We have a couple of questions from the students, so I invite Riccardo Capecchi to turn on video and sound and make his question to Dr. De Gregorio.
Hi, thank you for your presentation. In these lessons, we talked about the antibody dependent exacerbation as a possible risk in some specific condition of vaccination, for example, in dengue. And I've seen that there is a publication about SARS that hypothesize a sort of switching, polarizing of lung macrophages that are switched by low affinity antibodies and in a pro-inflammatory condition. I just ask if you have some, first question, if it's really an issue. And so if you know if there are some specific characteristic of the pathogen that can argue that they can induce an other response, and if you have a system to screen a possible antigen to eliminate or to reduce this I think this risk.
It's a good question. Thanks for the question. So I'm not an expert of SARS. I know that there is a risk for antibody-dependent enhancement of the pathology. I can tell you it's really something that has to be analyzed very carefully during vaccine development. For RSVF is the main question, because you need to know that in the 60s an RSV vaccine was with the classical method of whole kill formalin inactivated viruses was given to children and induced the increased death of children during the developmental phase. So for RSV, we know that a vaccine can do even worse than not being vaccinated. For dengue, it's the same. We know that you can have, it's very well known that if you don't have a robust neutralizing antibody against one strain, you can actually induce the infection from another strain. So it is a question that is extremely relevant. And that's why I'm a bit scared when people propose that you have a vaccine in 6 months or in 18 months, because this has to be analyzed very carefully. There is indeed, especially if you induce for viruses non-neutralizing antibodies, they just bind the virus by non-neutralize. What this antibody can do, of course, is to increase, there is a risk they increase the infection rate, or they do not really help the the immune response to be functional. On top of that, even CD4, CD8 response induced by vaccination have been associated to a potential immune exacerbation of inflammation. So we know that there is an exacerbation of inflammation with the COVID, and the cellular response to COVID is overwhelming and needs to be controlled. So what I'm saying is that, yes, indeed, vaccines have to be effective, but it's even more important that they're safe. I don't think that for COVID, it's a different story, because this vaccine will be used for billions of people. And the last thing you want to have is a vaccine that have a problem even in 1% or even in 0.5% or even in 0.1% of the population you give the vaccination to. So you need, by definition, large numbers, because this vaccine is going to be given to an enormous amount of people, to be sure that that you have a safe vaccine. You have a safe vaccines after vaccination, and that's easy in a way, because you just monitor the response after vaccination, and we have standard criteria. But moreover, as you are saying, you need to have a safe vaccines after infection. You need to find out if not only if it protects or not, but also if you're not generating some problem in some of the people, maybe a subset of people that are infected. So it's a very important question, and indeed needs to be addressed. For RSVF, having a prefusion, we know from some preliminary phase 1 data from NIH that the prefusion F essentially changed the ratio between IgG and neutralizing antibody, increased a lot more the neutralizing antibody versus general IgG. So most of the antibody you generate are neutralizing. What we hope is that by having more neutralizing antibody and less binders that are not functional, you limit the risk of immune enhancement. But you have to do a phase 3 and demonstrate that this is true. And this means thousands of people to be checked in the phase 3 study. Well, I think, I don't know if I have a lot of things to say. I don't know how much we can stay here on this slide. Maybe a last question, and then I will move on.
We have time. So there's a last question by Simone Frascolla.
Okay.
He has problem with a microphone, and it's a general question about which are the main steps that can be jumped or accelerated during vaccine development, and which are the risks if we speed up too much.
Fantastic. Well, why don't we keep that? So I show you all the technologies, and then we can really have a discussion at the end of the presentation on that, because some of the answer to the question will come in the next slide. So for this question, that is really what I ask the students to reflect on is this, and what could we do to prevent the next pandemic? Those are the two things that I think is important for the students to reflect on. This very same question, and also how can we get prepared for the next pandemic? So I move on. Antigens, we say many things about the antigens, but you know that the antigen alone sometimes is not good enough. The antigen without an induction of the innate immune response in a naive individual, it doesn't work. If we give an antigen to a person that has never seen this antigen, is not functional. You need to add an adjuvant, you know very well, to induce, to activate the dendritic cells, induce cytokines, MHC class 2, and co-stimulation to be able to induce CD4 T cells, and therefore help the B cell response, effector T cells, memory T cells. So all the, and I mean, you don't get CD8 here, but all the MHC class 2 driven responses, TFH, effector memory T, and B cells, really depend on the presence of the adjuvant, of the adjuvants in people that are naive. I have to say here, and I'm sorry not to have a slide for that, that if you give an antigen to individuals that have already experienced infection, like in the case of RSV or Staphylococcus, where we are all primed, the antigen is, even without adjuvant, even if it's super clean, is able to induce a response directly starting from memory B and T cells, and therefore the need for the adjuvant is less important. I'm not saying it doesn't impact, but you may have some vaccines Because you may say, why for subunit flu, we have a flu vaccine that is given to everyone and doesn't have an adjuvant? Because flu, you get exposed all the time. And even if you have a slightly different flu strain, you still have some cross-reactivity. So when you give the subunit flu to healthy individual, even without adjuvant, with one dose, is able to induce a response. When is the problem? Is when you have to give a vaccine to people that are naive, like in the case of COVID, like in the case of a completely new flu, like in the case of pandemic. So from our experience, for instance, H5N1, in the pandemic, or H1N1 even, in the pandemic that we had, the adjuvant was really essential to have a good response. H5N1 even more. So if you give an H5N1 vaccine, subunit, without adjuvant, you need 2 doses, and even after 2 doses, you don't have a very strong neutralizing titer. But if you had an adjuvant like AS03 or MF59, you start to see a neutralizing response after 1 dose, and you have a very good response after 2 doses. So it's really black and white. For COVID-19, guess what? You can inject all the proteins you want, but if you don't have an adjuvant, you are not going to have a response, because we really don't have any memory for COVID spike protein, except the one that already had infection, or the one that probably have experienced SARS, but is fortunately the minority of the people. So adjuvants are extremely important especially for the situations where we go to naive individual, and therefore for response to emerging infection, for sure, without adjuvants, you cannot have an effective subunit vaccine. We have in GSK several adjuvants that are licensed. Actually, GSK is the only company. I don't want to I mean, it's not to say GSK is good and the others are not, but indeed, GSK is in the front line of adjuvant discovery, and is the only company that has several adjuvants licensed. One is, and the adjuvants generally that we have are made of one component that we call classical adjuvants. Others call it antigen delivery systems, like aluminum, where the alum is absorbed, or liposomes, where the antigen can be incorporated, or oil in water emulsion is not really linked to the adjuvant. But those are classical adjuvants, empirical adjuvants. We know that they are important for delivery of the antigen. I've done a lot of work, many papers, I didn't send it to you, to show that these adjuvants are also eliciting cytokines, and therefore are also immune enhancer most of the time. Alum is a weak immune enhancer. Oil in water emulsions is also an empirical immune enhancer, acting on stromal cells, liposomes too. But I mean, we call them classical adjuvants because they have there is an element of delivery of the vaccine to the cells that is predominant. And then we have an immune enhancer that does genuinely deliver and added to increase in immune response. And generally, those are toll-like receptor agonists or agonists of inflammasome, caspase, these other things. So things that go to the pattern recognition receptor. One that we use is MPL, that goes to TOL4. One is a saponin, it's called QS21, it goes to inflammasome caspase, and on the interleukin 18 axis. And one in vitamin E is less known, but that is also an immune enhancer. So we by combining these things, we have developed an A04 that is aluminum salts, where you absorb at the same time the antigen and MPL. So it is a co-delivery of antigen and toll-like receptor 4 agonists on the same cells, and it's very efficient for a hepatitis B vaccine and also for human papillomavirus. It is licensed and is available. We had an A03 that is only water emulsion plus vitamin E, alpha tocopherol, that has been used for pandemic influenza, and is what I told you before, black and white. The vaccine without or with adjuvants, you have less response. You can use less antigens when you use the adjuvant. So also this notion of antigen sparing is extremely important, because if you need a lot of vaccines, one thing is that you have to use 100 micrograms of antigen, but if with the adjuvant you can reduce to 10 micrograms, it means that you amplify by 10 the number of doses that you can make. So it's important for the immune response, but also for antigen sparing. And we have data on A03 for H5 and from H1 flu that are extremely encouraging. The vaccine has been used, and there is a track safety record for that. And then we have A01 that is a mix of 3 components, liposome, MPL, and QS21. This is an incredible adjuvant because there is a synergy between TOL4 and everything that is activated by QS21 saponins, mainly inflammasomes. I mean, it's known that toll-like receptor and inflammasome synergize. And here, the combination of these three things made an extremely impressive zoster vaccine that is called Shingrix, and is used to eliminate the reactivation of zoster in the elderly. Just to give an idea, the viral vaccine was working 40–50%. This subunit vaccine plus AS01 is worth more than 90%. So it's really a big adjuvant effect. Also, the malaria vaccine that is tested now in Africa is an AS01 plus the malaria antigen. And I have one slide on TB, because TB is really an incredible story. So this is AS01, MPL, saponin, quite big structure, not small molecule. You're not there. It would be nice to have adjuvants made as small molecules. We have very difficult molecules to deal with that are these large molecules, saponins, MPL, that are embedded in the liposome. Is used for these candidate vaccines. We had this the results 2 years ago now of this phase 2 proof of concept trial, where by using Th1 epitopes from tuberculosis, that is called M72 candidate. Those are epitopes that are known to be induced by tuberculosis in human, plus the adjuvant system 01, we could get for the first time 54% efficacy in reactivation of the TB in infected people. It's the first time that there is proof of concept for a tuberculosis vaccines, and is really due. What made the difference here is really the S01 adjuvant, that is unique ability to induce a very strong Th1 CD40 cell response. So adjuvants are important for improve efficacy in naive subjects and in the elderly. For zoster, you have to consider that the zoster vaccine is given to people more than 50 and even a lot more than that. I mean, you all know I'm 50. I know that my time was over 10 years ago. Most people don't understand how fast immune senescence starts. It's quite bad, but I mean, it starts pretty early. When you're 65, the tendency is to respond less to vaccination, even if you've been primed. And especially in this population, in the elderly, the adjuvant is extremely important. So think about COVID, think about the target population. You can imagine how important it is to have a strong adjuvant for any vaccine response to COVID. People are naive and are elderly. You really need an adjuvant if you want a vaccine to work. You have enhanced cross-reactivity. I didn't say that, but on flu, the data on flu shows that with the adjuvant, you have more cross-reactivity in flu for mismatched strains. You have an acceleration of protective response. For HBV, we have seen that you can limit the number of doses with an adjuvant. There is another adjuvant, not from GSK, CPG, from another company that shows that you can have, for HBV, a reduced number of doses. You don't have to use 4, but you can use 2 doses to get protective response. So you accelerate the protective response because you need less doses, essentially. You have reduced number of doses, and very, very important, reduce antigen dose. Dose sparing is key because I don't know who's going to make the first vaccine against COVID, but manufacturing enough antigen for more than 1 billion people is an unprecedented challenge. So if with an adjuvant, you can reduce to the minimum required by tenfold, this really address the medical needs. Now, I'm not going to go in details on adjuvants. There is a lot that can be said, but maybe we can leave it for the question session. What about the approach that takes together protein vaccines and adjuvant for emerging infections? So it is very promising. It's a combination of 2 things: structural design. In COVID, is the pre-fusion spike, that's the structural design element, and the adjuvant to increase probability of success. I believe that this is really the vaccine at the moment with the highest probability of success, because we know that neutralizing antibody exists, and we know that if you have the right antigens plus the right adjuvant, you probably will induce this antibody in humans. So this is really an established proof of concept. For other targets, I think that we can hope that this is going to work for COVID as well. I'm pretty confident that this is going to work for COVID. It works in preclinical data. We will see. It is possible to produce millions of doses, yes, but still, you have to find the right antigen production method to do it. CHO, quite difficult. You have to find something else because it's a complex manufacturing process. We have licensed the Shingrix vaccines now 3 or 4 years ago. We still don't have the doses that are needed in the world. And imagine for a vaccine that makes a lot of money, like Shingrix, the pressure that we have in the company to increase the availability of this vaccine. So it takes years to have enough production plants and increase the number of doses for a vaccines made in CHO cells. And so I'm really worried that the problem for this approach is, are we going to have enough vaccines for everybody if everybody needs vaccination? Because the scale up is really a problem. Each time you have a new protein, it's not a platform technology. Each time you have a new protein is a different process for production, for purification of the protein, and for formulation. All the process is new. So you have to establish a new process for each new vaccines, and you have to make a production plan that is tailored to that. You can use old production plants, yes, probably yes, but there are adaptations you need to do. It's not so easy to have enough doses. That's why I wrote slow. It's slow from the concept of vaccine to have enough doses for everyone. In GSK, what we decided to do is there are many companies that have proteins pre-fusion that seems to be the right antigens. We all hope is going to be the right antigens. But very few companies have licensed adjuvants with the track safety records. We have it. So what we decided to do is to collaborate with all the companies that are making these antigens and provide our adjuvants for R&D, for research and development of new vaccines. In particular, we had a big collaboration with Sanofi. Why with Sanofi? Because Sanofi knows how to make eukaryotic proteins with a different system that is not the classical CHO cells, but is using baculovirus. It's a system that they already use for flu. And so it's already used for millions of doses. So they have the possibility, because they have big plans for making protein, to scale up, if the vaccine works, rapidly to many, many doses. And if we provide the adjuvants, I think it's a good synergy to have a good vaccine. That's the only thing I wanted to say. So that's the approach that we favor at the moment in terms of probability of success. It's not the fastest way. Before you have GMP material to go to humans, it takes time. So you have to make GMP material in CHO. We still don't have it, in fact. This needs time. In fact, if you look at the vaccines in the paper that I gave to you, and we can go there later, on the vaccines that are already in clinical development, they are either inactivated vaccine, classical Pasteur principle, RNA vaccine, non-replicated vectors, or replicated vectors, not subunit vaccine. The subunit vaccine takes more time. And so it takes more time before you can test it to human. What are these other things? So viral vectors and RNA vaccines. This is the last part. What do you prefer? I asked the chairman, I stop or I finish my presentation, then we have a long discussion.
I guess that we can go on, and at the end we have the discussion. Thank you.
So I spoke about the subunit vaccine and adjuvant, especially for viral vaccines. There are other options. And as you can imagine, those are the ones that are faster, but and one is viral vectors. One you heard for sure about is RNA vaccine. I'm sorry, I didn't wrote here DNA, but I can talk about DNA as well. So DNA vaccines, viral vectors, and RNA vaccines. Let's start with viral vectors. So there are several viral vectors you can make by using, especially adenovirus is the one that's more used. That can be adenovirus from human or for chimps, and I can tell you why. Vesicular stomatitis virus, alpha virus, pox virus like MBA. Those are the typical viral vector use. What is the concept? You make a vector, you engineer the vector to express the gene of interest. Generally, you remove the proteins that are needed to replicate, And therefore, after the infection of the virus to the cell, the virus does not replicate, does not form another virus, but just express your gene of interest. The molecular biology is different according to the different vectors, but for the non-replicating vector is always the same. You give vectors that introduce this piece of genetic material, DNA or RNA in the cell express the gene of interest. Or you can have replicating vectors where they induce, especially if you already have a live attenuated vectors, you can engineer this live attenuated vector that already exists to express the protein. And in this case, like in the case of VSV, it's a live attenuated vector. You have an infection that is self-limiting and you express a lot of antigen. Proof of concept they work, yes, there is a licensed Ebola vaccine using VSV vector, replicating vectors, and these are chimeric yellow fever vaccines that be licensed for dengue. And a lot of phase 2 and even phase 2B showing that with adenovirus you can induce a good response, or with pox virus, MVA, you can induce a good response. So we have evidence that adeno, MVA, VSV are effective in inducing antibody, CD4 and CD8 responses, as I said at the beginning, right? So it's MHC class 1, MHC class 2, you get everything. What is the advantage? The advantage is that those are kind of platforms. So if I have a process to express an adenovirus, and I have already a fermentation protocol, purification protocol, and production protocol, if I change this adeno, instead of one gene of interest, I put another gene of interest, it's not a big change. So the process is ready. I just change the gene and I have the process ready, right? It's not like having a CHO protein, a new CHO protein that requires new conditions for processing, especially new condition for purification, because as you know, each protein needs to be purified with different columns with different processes. So, and that's why the adenovirus and replicating vectors. So there is in clinical trial for COVID, there is already a type 5 adenovirus and then cheap adenovirus. That's why they are ready to go, because the GMP production process was ready, it's there. We can use the platform. That's why we call it a platform. We can use the platform knowledge that we have. We plug in the gene of interest and we can have GMP material pretty soon. That's what they did. And that's why we have this vaccine first. It's good, nothing against that. I can tell you what is the limitation of these vaccines. One limitation is that, for instance, adenovirus, if you have been exposed to adeno, and many of us are exposed to adeno, you have antibody against adeno, and so the response to adeno is very limited in people with pre-existing immunity. That's one problem. So that's why the chimp adeno has been developed to avoid pre-existing immunity. There is some cross-reactivity, even with chimp adeno, you have some pre-existing immunity, but it's pretty good. So let's say there is one adeno type 5 from China in phase 2. Well, the risk will be that some people will not respond very well to the vaccine because of persistent immunity, because they've been exposed to adeno infection. There is a chimp adeno, better chance to have a response of the vaccine because it comes from chimps and there is just some cross-reactivity. But what is the additional problem also with chimp adeno? We know that after the first dose with chimp adeno, you generate antibody against your gene of interest, but you also generate antibody against your structural protein that are present in your vaccine. And therefore, on the second dose, the response is not that good. If these vaccines work with one dose, fine. If it works with 2 doses, well, very difficult to have this vaccine effective for COVID. My expectation is my point of view. People are naive, probably will need 2 doses. So we will see. I hope that if after 1 dose you have protective neutralizing antibody, but I suspect that the response of this vaccine will be rather weak because chimp will respond after the first dose. It will give some level of neutralizing antibody, but it will be not possible to boost with the second dose. And therefore, we may have some limited neutralizing response, maybe not lasting for many, many months. I mean, this is my opinion. And it's like, I'm telling you really what are the limits of the vaccines that are there. I hope that they're going to work. They're in phase 1, it will take time, but there is a risk that with the vectors, you may have a problem with the, because you generate antibody that neutralize the mechanism of action of the vector itself, because they recognize the vector. I think it's pretty clear, everybody knows. How can you cope with that? Well, you could use an adeno prime MVA boost, for instance, then it's fine. But at the moment, I don't see an MVA, an adeno prime boost strategy in phase 1. So this will come later. So here's is a platform technology, fast discovery, good antibody and cellular response, but anti-vector immunity. And the other problem is also very complex manufacturing. So even if it is a platform, you still have to make the vaccine vector in a cell culture. It's a complex biological process, difficult to make a large number of doses, difficult to make a stable vaccine at room temperature. Sometimes have to be lyophilized. I mean, there are issues related to the fact that this is a complex manufacturing technology. So you have a fast entry into phase 1, but then for the scale up and made a lot of doses, they may have a problem. So what else is rapid is DNA and RNA vaccine. I don't have DNA, I have RNA, but DNA I can talk about it because there is indeed a DNA from Inovio that just finished, is doing phase 1. So what is the problem with DNA? So DNA, essentially what you do, you make a plasmid and you inject the plasmid. The plasmid that of DNA with your gene of interest under a strong promoter. In animals, works pretty well. You can have in mice a very strong antibody response with DNA. When you move to large primates and to humans, well, you know, it has been very disappointing. In fact, that's why we don't have a DNA vaccine, despite the fact that the first DNA vaccine in preclinical model was more than 20 years ago. Yes, a lot more than, I think in the 90s, in the early 90s. So we're talking about more than 30 years ago. We don't know why. We don't know why in mice it works and in human it doesn't work very well. But that it's a matter of fact. It's like cross-presentation with the adjuvant. In mice, we see that MPL induced CD8. We move to human, you don't see CD8 anymore. So there are things that the animal models cannot predict and do not translate. So this is one of the cases. So DNA can be used in human, but you have to help them to work. And the way this company, Inovio, that is in phase 1, is using to help DNA to work is an electroporation. So it's a device that through an electroporation in the muscle make the DNA work. Well, I haven't done it myself. They say that the electroporation is not that painful, but still you can imagine, I mean, it's not just a simple injection, it's an electroporation. So can be used for billions of doses, can be administered with electroporation, I don't know. But I mean, it's there, it's in phase 1. With the electroporation, it seems to work. Can be scaled up, it's easy to make a plasmid. The questions are potency. Is it potent enough even with electroporation? And is it possible to give the electroporation to everyone? What is the impact of electroporation instead of a simple injection to a large population? That's the big question there. But doesn't generate anti-vector immunity. You can boost as many times as you want. So those are the advantages. So you don't have anti-vector immunity. You can boost, but is weaker than viral platform in terms of immune response, needs electroporation, and maybe too weak. We will see. Times will tell. But it's a promising approach. I'm not saying it's not promising, and it's in phase 1. The reason why RNA vaccine have been, we started to work as well on RNA vaccine is really because we wanted a nucleic acid vaccine without anti-vector immunity, easy to make, a platform technology with more efficient than DNA and without anti-vector immunity. And that's why several years ago, people started to work on this RNA concept. Now there are already, how many RNA? I mean, there is one RNA from Moderna in US that is finishing phase 1, and there are many RNA platforms that are coming to phase 1. There are two different types of RNA vaccine, mRNA or self-amplifying RNA. And you can divide the mRNA vaccine in 2 categories, one that use natural nucleotides and one that use chemically modified nucleotides. So I spent a few words in here. mRNA is essentially an RNA made of cap, 5' untranslated region, your gene of interest, 3' untranslated region, and poly-A. So it's very similar to a natural RNA. And this is true for natural and chemically modified. You can make this RNA from a plasmid template in an in vitro transcription. It is a platform technology. You can make it easily. If you have a new emerging infection, in what Moderna did is in 42 days, they went from the design to the vaccine ready for GMP. That's as easy as it is. Because really, I mean, the process doesn't really change if you have to produce one gene or another. It's a single strand RNA, mRNA, and it works pretty well. What is the problem? mRNA is not very efficient. You need to limit the interferon response. And some company use natural nucleotides and try to limit the interferon response by working on the sequence and the secondary structure of the mRNA. So they try to limit hairpins, they try to limit sequence that are strong and known to be strong interferon inducer. And by working on the sequence of the mRNA, they can then have a good response. For chemically modified nucleotides, it's different. They use these chemically uridine nucleotides to reduce the interferon response. If you reduce interferon response, you have a better expression of the antigen through mRNA, and it helps. Both needs a very good antigen delivery systems, and nowadays they use some cationic liposomes. The self-amplifying RNA, it's an interesting story because it derives from the RNA from RNA viruses, devoid of structured protein. What is the advantage versus the mRNA technology? Is that starting from a very low dose, because they amplify in the cells, you can have a very good response by using less RNA. But there is proof of concept preclinical that both mRNA and self-amplifying mRNA works pretty well, better than DNA electroporation, and a very low dose, provided that you have a good RNA delivery systems. This is a scheme of the mechanism of self-amplifying RNA. And I will spend a few words because that's what we have. We are working on self-amplifying RNA. It can be formulated with the delivery system that can be an oil and water emulsion or a cationic liposome. The cationic liposome seems to work better. You have a sequence that has your gene of interest and viral non-structural protein from alpha virus. When this RNA goes into the cytoplasm, you have this positive strand RNA with cap, 5' UTR, 4 non-structural protein for alpha virus, your gene of interest. What happens is that these 4 non-structural protein, yellow here, gets translated, and they start to make a lot of minor strand RNA that have your gene of interest. And from this template, minor strand that is like 1,000-fold more than your input material, there is a transcription RNA-dependent RNA transcriptase that transcribe your gene of interest, and then this gene of interest is further amplified and translated. So from one molecule, you have thousands of molecules of gene of interest that are made, and therefore you have a very sustained expression of RNA. It's not clear whether it goes into the endosome and then released, or is the direct fusion, probably both. In any case, this RNA is eliciting interferon through TLR7, TLR3, and through innate immune response, IMDA, RIG-I, PKR, so the cytoplasmic sensor of RNA. But despite inducing interferon, this is able to induce a strong response because of the potent replication. Just to give you an idea, if I use for the same gene, luciferase, a self-amplifying RNA or an mRNA with the same delivery systems, you can see the difference with the 0.1 microgram of modified RNA. That's the expression I have in terms of bioluminescence. With the self-amplifying RNA, it's called SAM here, I get logarithmic scale, more expression of antigen, and the antigen expression lasts for 20 days. So what we think is that with the SAM RNA, we have the opportunity to have a single dose vaccine and to use very, very small amounts of RNA and make a lot more doses compared to mRNA. But both strategies are valid and they're both used for COVID. This is one paper. The first paper we published on SAM is from 2012. You see how is it made. The RNA is inside the liposome. The liposome contains lipids, some cationic lipids that, of course, are important for formulating the minus negatively charged RNA, and cholesterol, and the RNA is inside, protected from degradation. If you use this formulation, here is an example. You have VRP is the alpha virus vector, where the gene is delivered through the vector containing the same replicon. Just very close is 20 micrograms of DNA with the same gene of interest electroporated, and you can see that 20 micrograms electroporated have the same effect of 1 microgram roughly, or even 0.1 microgram of, actually it's 1.1 microgram of LNP-RNA. So if you use a fraction of microgram of LNP-RNA, you get the same antibody response that you get with 20 micrograms of DNA electroporated. So it's an order of magnitude more potent than DNA. We tested that already in mice and non-human primates compared to standard vaccines. Here you can see that the vaccine gives a good rabies neutralizing antibody response, even if you go to 0.00015 micrograms. So there is a potential really to have a very effective vaccine, a very small dose. Next slide, you can see these vaccines in terms of CD4, CD8 response compared to a whole kill inactivated vaccine. Well, as I told you before, whole kill inactivated vaccine doesn't really give you CD8 response, but you can see the RNA vaccine gives a very strong CD8 response and also very good CD4 response. Here is non-human primates, a protein from CMV. You can see the difference between 15 micrograms of GB proteins made in CHO formulated with our best possible adjuvant, that is S01, compared to 75 micrograms or 3 micrograms of the same protein in SAM. You can see the neutralizing assay on epithelial cells, the antibody response is pretty similar. The CD4 T cell is pretty similar, but with the SAM, you add the CD8 response that cannot be made even with the best protein, with the best adjuvant. So the preclinical profile is very, very promising. There is the potential to make many, many doses of vaccines, and also to have a very robust CD4, CD8, and antibody response. Manufacturing for RNA is pretty easy. You make it, as I told you, it's a DNA. Then you use a cell-free RNA synthesis systems when you need to get nucleotides and the RNA polymerized, essentially, and the capping enzyme. Purification is pretty easy. It's a synthetic vaccine. It's not a biological, so with all the advantages of it. And so it's easy to get the drug product. Moderna, to make an mRNA, it took 42 days from the sequence of COVID to the GMP material. It's unprecedented. It's really super, super fast. And this is what I said in terms of the opportunity for RNA in terms of response to pathogen. There is a faster GMP material and potentially also ability to scale up. This, I mean, how can we scale up depends on the dose you need to use. So platform technology, fast discovery, good antibody cellular response, fully synthetic, no anti-vector immunity, potential for millions of doses. You may think we have the solutions. Yes, but there is always a but. There is no RNA vaccine license yet. Proof of concept for safety of RNA vaccine is not established. Also for efficacy in humans is not established, and it's never been produced in commercial volumes. So risk is down the road. They may have problems when we scale up. So we don't know whether this is going to work in terms of scale up. That's to be very clear. So very promising, but the proof of concept has to be built. I had a few slides on bacterial vaccine technology, this GEMMA. I think I can skip that because I think it would be more, because it's not used for viruses, and we are focusing more on viruses here. And what I propose to do is to move to the question and answer. Maybe what I can do is to open the list of vaccines that are in the clinical trials. Let's see what I can find it. No, it's not here. To stimulate the discussion, let me check.
Is it the same table that you sent to us by email a few days ago?
Yes, it's the last table from, I think it's WHO. It's,
okay, maybe I can.
I have it.
Okay.
I have it. I have it here. Here it is.
Okay.
So I keep this here. This list on the first part, the candidates that are already in human vaccine evaluation, as I told, are the platform technology, not surprisingly, vectors, DNA, inactivated. It's not a platform technology, but it's easy to make the whole inactivated because you just make the virus and then you inactivate it, beta protolacton. And I have a paper, if you want to see the preclinical data of this inactivated
Dr. Gregorio,
vaccine.
Can we share the file with the students? Can I send this file to the students?
Yes, this is something that can be shared. Yes, it's
okay.
not. It's publicly available, and I also have a paper that shows for this vaccine. If you are interested, we can go through the data. This vaccine from where is inactivated Sinovac, inactivated plus alum. There is a paper, and this, as far as I know, is the most complete preclinical package that I've seen, which shows what the vaccine does, even in a challenge model with non-human primates. So if you want to see some preclinical data of a COVID vaccine, I can open this this paper for you. Having said that, open for question. If you want to talk about COVID, happy to do it. If you want to talk about anything else, I'm here.
Thank you for this really wonderful lecture, very clear, and thank you again. So
let me just close the window because they're making works here.
Okay, I have sent the file in the chat to students so they can download them. So there are a couple of questions by Francesco Castagna. So Francesco, please.
Yeah, here I am. Right. Can I go on with question?
Fine with me.
Okay, great. Thank you. Thank you very much for your presentation. It was very, very interesting. I have a couple of questions. One regards the use of ex vivo dendritic cells as an auto transplant. So if you are aware or if you know that in study the use of dendritic cells ex vivo primed with microbial antigens in order to have like kind of vaccination and reimplanted into the patients or volunteers.
Yes. Yeah, there is a vaccine, it's called Provenge for cancer that tried this way to take the cells from the patients, including dendritic cells, prime with the tumor antigens and then give back that to the patients. I think it was prostate cancer. There is proof of concept that can help, but I can tell you it's very difficult to provide a vaccine made this way in a consistent way to patients. And the logistic operations, it's really, really difficult. So it's for sure for cancer has been considered, but for infectious diseases is not something you would consider. But there is proof of concept. Indeed, it's one promising approach for very difficult chronic disease. But you can imagine having a consistent process to do it is not that easy. You should. There are issues with that. But it's possible that this may be for vaccines for very difficult chronic conditions. For sure, cancer, maybe some infectious disease could be one approach that can be made. We're having a process in the hospital to essentially work on the cells from the patients and then re-inject the cells. It's something that is done with T cells now for cancer, you probably know, with the T cell approach. but it's not easy. I can tell you, the big challenge is how do you provide that on the large scale in a consistent way.
All right, thanks. And the second question is, there's a few evidence that aggregated proteins like monoclonal antibodies are more immunogenic than the monomeric version. That's why some people, some patients getting monoclonal antibodies develop an anti-drug antibody response. And it's, I mean, there's an idea of disaggregation being one of the main reasons for the anti-drug antibody response. Are you aware of anyone working on a vaccine made of aggregated subvisible particles of microbial proteins, antigens?
Yes. So essentially, the theory is, as you know, if you have antigens that are in a aggregation or repeated array, you have a better chance to cross-link B-cell receptors. So the theory is, if you have it, it's probably you increase the ability to cross-link the B-cell receptor, so you have a better impact on B-cell receptor. On top of that, if they are particulates, there is a better uptake by the dendritic cells. So you have these two events that can synergize for the immune response. One more to presentation to T cells, one more to direct engagement of B-cell receptor. There is evidence that this is the case. So if you have multimerized antigens, they work better. Now, how do you achieve that? One way to achieve that is VLP. You have antigens that form viral-like particle, extremely potent antigens. HPV, HBV vaccines are very potent. They last for the effect lasts for years. And what some people believe that the fact that they're multimeric in a VLP is one of the reasons why this is the case, because they're particulate and because they're multimeric. You can achieve the same by using an antigen delivery system. For instance, why the alum works? Well, probably for the same reason. Antigens are aggregates adsorbed to alum, and then alum forms some sort of microcrystals in the body where the antigen is absorbed. There is an increased uptake of the antigen because it's in a particular nature, and there is maybe an increase, we don't know for sure, but there may be an increase cross-linking of B-cell receptor because it's absorbed to alum. We prefer to have a way to multimerize the antigen that is controlled. Aggregation is difficult to control. So in theory, may work, but having a vaccine that consistently aggregate the way you want and does not precipitate actually, but just aggregate in the particulate that you want is very unlikely to work. So we try to avoid aggregation, and we try to rationally multimerize proteins in the forms that improve uptake by dendritic cells and antigen-presenting cells and B-cell cross-linking. That's the response. You are right, we do it, but not through aggregation, through a more rational design of the vaccine.
Thank you very much.
So thank you. I would like to remind people who are following us on YouTube that they can post their questions on Twitter. So just please use the hashtag SOI2020. So there's a question from the audience from Erika Parisi. So I invite her to turn on video and sound and make a question. Erika, are you here? No. While Erika connects, I would ask Lorenzo Cuollo to make his question, please.
Hi, can you hear me?
Yes.
Yeah, thank you for the wonderful presentation. And my question was about the safety of self-amplifying mRNA vaccines that you explained to us. Do you think there is even the remote possibility that this kind of vaccine might reactivate latent infection, or maybe even, let's say, mobilize retroviral sequences in our genome, or so maybe, I don't know, causing any sort of problem like transformation of cells and so on?
So this vaccine is an RNA replicant, does not integrate. First, doesn't integrate. That we know about that. There is not a retro transcribing is RNA-dependent RNA polymerase. So there is not a DNA form. Doesn't have proteins that are shared with the virus that integrates and may be reactivated. So I think it's very unlikely. It's not based on lentiviruses or things like that. So it should not. So I don't think that there is any safety concern or reactivation of what is integrated in the genome, because it doesn't act on DNA. So I think it's very unlikely. But I mean, it's a good question. I think we should probably better assess that hypothesis. But I don't think that there is such a risk. The safety concern may come more, I believe, from reactogenicity. So it doesn't integrate, doesn't reactivate things in the genome, but is an RNA in cells that stimulate an interferon response. So so we are testing this in phase 1. So I cannot talk about the data because we still haven't seen the data. But my expectation is that what will be key for self-amplifying RNA is to get the good therapeutic windows for reactogenicity. So don't overshoot with RNA. We should give just the RNA we need, and it will be good to limit to 1 or even less, I mean, a few micrograms or even less than that, so that we get a robust response without having a strong inflammatory or interferon response in the body. So I expect potential for fever, for pain, but it's still reactogenicity. I don't expect big safety concern. Now, everything needs to be done, checked carefully, even long-term safety or reactivation of viruses will be for sure something that will be monitored in the trial. But I think that the key risk is that with RNA in general, is that they're too reactogenic. People get fever and then they don't want to get the second dose, for instance, or things like that. Because RNA is a very active molecule in the body, that's for sure. You think about how many RNA sensors exist, and our body, especially the innate immune response, has a lot of sensors for RNA because of the presence of RNA viruses. The interferon response can be something that has to be managed here. That's what I believe.
Okay, thank you very much.
Thank you. We have a question by Junbiao Wang. So please, Junbiao, can you say your question by yourself?
Okay.
Thank you.
Okay, thank you. Thank you very much for your talk. And Max asked a general question, because we are, could you please talk something about the phage-based vaccine? Could we use phage as a vector or modify the phage to as a vaccine?
Well, I'm not an expert on phage vaccines. I know that phage have been proposed for antimicrobial resistance as a way to kill or target bacteria. I think that there is potential for phages to, you're talking about bacteriophages, right? So to use phage to address some bacterial diseases. I think that there is such a potential. Where is the risk is that even when you inject the phage, you will generate an immune response against the phage. So you cannot avoid that the body will be able to neutralize the mechanism of action of the phages. But otherwise, yes, we could think about using phages to insert genetic material into bacteria that then can be targeted in a way or another. Through a vaccination or inject something that directly kills the bacteria. So I think that it's an interesting field. I'm not an expert there. This is not something that we do at the moment, but I see the risk of the immune response to the phage as a major limitation.
Okay, thank you.
Thank you. So we have a question by Mirela Kuka, please.
Hi, Enya. Thank you for the presentation. So I just want to know your opinion, your take on why do we struggle so much in having T-cell vaccines, not good T-cell vaccines, because at now there are not at all, if I understand correctly.
Well, the TB slide that I showed is not inducing antibodies. The TB vaccine that has been designed with the T-cell epitope and AS0, A01B, A01E that you show in the slides, it acts through CD4 T cells. It's pretty obvious. So it's in phase 2, it's not licensed, but it's very promising. And then it's difficult to demonstrate that the CD4 and CD8 contribute to the effect of the vaccine. But I think that even if in the vaccine that we use, there is indeed a CD4, CD8 response. I don't want to be misunderstood. It's the evidence that is lacking, but I think they do work to CD4, CD8. So I think that the live attenuated vaccine, they may work also because of the CD8, CD4 response. I think that the Shingrix vaccines works essentially because you have a very good Th1 response and antibody-dependent cellular cytotoxicity, also mediated by NK, but can also be mediated by CD8 T cells, CD4 T cells, sorry. So in other words, it's difficult to demonstrate if you have vaccines that work through antibodies and T cell. The antibody correlate has been identified in some cases. In many cases, it's more difficult to demonstrate that CD4 or CD8 that there contribute. But I think they do contribute.
Okay. Do you think there are any technical limitations in demonstrating that one or the other? For example, I don't know, antibodies you can purify,
Yeah, sure. No. It's much more difficult to demonstrate because with antibodies it's easier. That's for sure. With playing with the cellular immunity is much more difficult, and demonstrate that is also much more difficult. But I mean, in animal models, it's been demonstrated, right? We have evidence in animal models that there is a cellular contribution, even for flu subunit vaccines. You can do experiments in mice and show that the CD4 helps. For other bacteria as well. The demonstration is more difficult in humans. That's the only point. I didn't want to say that the T-cell response is useless for the vaccines that we are developing.
No, absolutely.
I'm just saying that it's more difficult.
In fact, I know that also for HIV trials, there are many trials that are pointing towards the T-cell response, CD8. When I say T cells, I intend the cytotoxic.
Yes. Well, one thing needs to be said, that vaccines that were designed to work exclusively through a CD8 response so far didn't work. They address very difficult pathogens like HCV and HIV, for which we have an issue with many different obstacles, but they didn't work. So the vaccines that are out there that are licensed, as far as I know, all of them, they have a functional antibody response, and they may have the T cell contribute. But the vaccines designed to induce only T cell response, except for TB, for the first time we see some effect in this only T cell response, but most of them have failed, which is an interesting, probably it's difficult to maintain, while for antibody, probably you guys, you know more than me, I'm not an immunologist by background. I've been involved in immunology since 20 years, but I'm not. But the impression is that you have, when you induce a strong B cell memory, this can really last for many years. And a systemic antibody response tends to go in the district where it needs to go. So if you have a good systemic response of antibody, there is exudate in the lung, there is exudate in the mucosal level, that may be enough everywhere. Having the right T cell response in the right place and sustain to respond to pathogen is probably more difficult than for plasma cell B cell response. That's my impression that I have. So you have to get the antibody are made and tend to diffuse everywhere enough to protect even from mucosal infection. T cell it's more difficult to have a vaccine that induces the right T cell at the same time at the same place. That would be my guess, but maybe I'm wrong.
Okay, thank you.
So Ercilia, please make your question.
Hello, thank you for your very clear presentation. My question is about the self-amplifying mRNA vaccine. Once the immune response is activated, couldn't be put into the sequence also sequencing to stop the expression? I don't know if anyone thought about this. And the mRNA sequence is also subject to mutation during time. So this could be a big risk, I think. And another question is more general, and it's which vaccine do you think is the best choice given all the information that you gave us?
For COVID or in general?
Yes, for COVID, yes.
For COVID. So the RNA stops by itself. It's self-limiting because the cell, after rounds of expression, will eventually stop making the RNA and die. So it is a self. So we did not put something that terminate the expression. It can be, I think the colleagues in the US that work on these vaccines, depends on the protein, they try to find way to rationally determine for how long the vaccine is expressed by working on the sequence. But at the moment, it is expressed for as you see, for more than a week. It's like even 2-3 weeks. But eventually, then the RNA disappears. It's not there anymore. So in these 2 weeks, now is there a chance to have some antigens that are not the correct sequence anymore? Maybe yes, but I don't think I don't see a safety concern there. So you may have, what you're saying is that during this replication and during the expression for 1 or 2 weeks, some of the antigen may not have the original sequence. It's possible. We have not checked for that. What we saw is that consistently you get functional antibody for RSV, you get functional antibody for CMV. There is not a reduction of functional antibody over time. So I don't know if this is really an issue.
Okay,
not for safety.
Sorry, thank you very much.
And for the best approach for COVID is a $1 billion question. I don't know. I think that if we will have time to make the vaccine, we will not consider the time element. Maybe it's easier because I think that, as I said, in my opinion, the vaccine that has the highest probability of success is the subunit vaccine. Well, I mean, a good pre-fusion protein with an adjuvant, because we know it works for other vaccines. I think it's the one that is going to work. The only problem is that may be too late. And whether we still need a vaccine next year, actually, I don't know. We may have the vaccine where we don't need it. RNA is good to try also this fast approach, because the time element is key, and anything that can come faster should be explored, but we don't know whether it's going to work. So I don't know. Honestly, I think that there is a good chance that the vaccine, this vaccine is not that difficult. What I mean is that from preclinical data I've seen, even an inactivated vaccine with alum can induce neutralizing antibody and protect non-human primates from intratracheal challenge, which is pretty good. I mean, there are some pathogens for which you don't see complete protection in animal models, like HCV, HIV. This is not one of these very, very difficult viruses that mutates like crazy, and the vaccine is really difficult. It's not one of these viruses that evade the immune system, so you get chronic infections or repeated infections, we don't know, but at least you don't get chronic infections. So in principle, it should be possible to make a vaccine. The body is able to terminate the infection, so the vaccine should be able to prevent infection. That's my point of view. If neutralizing antibody are key For prevention, it looks like even the inactivated virus can make it. So it's promising. How many doses they can do, and I don't know. RNA may work, but the one that for sure will work, if the assumption is the neutralizing antibody are the correlate of protection, is the protein vaccine plus the adjuvant. We may end up in a situation that one option, one producer, one manufacturer could not be enough to immunize everyone. So we may end up in a situation where we have, for the first time, multiple vaccines trying to cover as many people as possible, and maybe some will get an RNA vaccine, some will get a protein adjuvant vaccine. This is, I don't know. So it's good to have many players. There is hope that a vaccine in this case may work, but we have to be careful. We have to monitor safety, and the assumption is neutralizing antibody makes the job, but we have to verify with an efficacy trial that this is really true, and also that we don't have any enhanced pathology due to vaccination. Those are the things that we need to see. But there is a chance that inactivated RNA and protein adjuvant vaccine in different timelines may work. I'm less optimistic about the viral vectors because of the reason that I say that after one dose you cannot boost, and some people have anti-vector immunity. Those are the ones that, and I'm also less optimistic for DNA because I think it gives a weak response, and I don't think electroporation is really something that I would recommend for a mass scale immunization. So, but RNA, if it can be scaled up, protein adjuvanted if it's fast enough, and inactivated virus even are promising.
Thank you very much for your answer. Thank you.
Thank you. Riccardo Capecchi has another question. Please, Riccardo.
Sorry, thank you so much. Sorry, it's a super interesting argument. So I used to work on primary and secondary immunodeficiency. So when I talk about immunodeficiency, we evaluate the response to vaccination also for clinical criteria. And there are many types of immunodeficiency. So some patient doesn't have a proper response, a proper immune response, mainly B response. And whoever actually the line guides are to vaccine, of course, these patients with not attenuated vaccines in order to induce some sort of immune response in these patients, very low B response, but we hope that a T response still will be. And I just ask if, because I've see that you develop a COPD vaccine, or it's in order to develop. So do you think it is in order to project a tailored type of vaccine target for a very specific group of patients, for example, in the elderly or in immunodeficiency patient when you don't have a sufficient immune response, so probably with more adjuvants or different adjuvants, or that induce a different CD8 response?
Yes. That's a good question. So the vaccines specific for immunocompromised are not developed. So we have vaccines for everyone. I mean, the principle of vaccines that you give it with a large population, and you hope that work for everyone. But indeed, as you say, I mean, we can use vaccines for some immunocompromised. The important thing is to do a dedicated trial with immunocompromised. For instance, for the Shingrix vaccines, we are testing the Shingrix in immunocompromised because, indeed, in immunocompromised, reactivation of varicella zoster is a big problem.
Yeah.
And so we are testing whether the vaccine works, and we hope that works. The company in general don't start a vaccine program dedicated to immunocompromised. It's a good suggestion. I haven't seen that. Generally, what we do is first we do the vaccine for healthy, or in the case of COPD, for people that have COPD, and then as a second step, you check whether they work also for immunocompromised. I've never seen a vaccine that was tailored to immunocompromised, trying to find the magic component of the specific adjuvant for immunocompromised. It's a good question. I didn't think about that. there are regulatory risks, there may be commercial risks, maybe that's the reason why it hasn't been done, because it's too risky from a regulatory safety standpoint to start to address immunocompromise. We'll have to start from healthy anyway, but it's a good question. I believe that some of these adjuvants that we found make the difference in elderly may also make the difference in immunocompromised. But as you say, it depends on the type of immune deficiency that you have. In some cases, maybe the right adjuvants, in some cases not. But it's a good question. I think that to the question, could you design a vaccine that or an adjuvant that is specific for immunocompromise? It's an interesting question. So, and at the moment, as far as I know, we don't have such a tool in our toolbox.
Thank you.
I'm sorry, the answer was fragmented, but I hope you understood my answer.
So we have one last question by Maria Guevara.
Yeah, can you hear me?
Yes, we can.
First, thank you so much for your presentation. I have one question regarding the administration route of these lipid nanoparticle-based RNA vaccines. I am aware that some, for example, German company BioNTech that is also working on this technology uses an intravenous administration route, and they claim that this way they can target dendritic cells that potentiate the immunogenicity of the vaccine. But also, I know that these self-amplifying mRNA-based vaccines are often given intramuscularly. And also there are some preclinical data showing that they achieve good immunogenicity by giving intradermal or subcutaneous. So do you know if these differences in administration route may impact the immunogenicity of these lipid nanoparticle-based RNA vaccines, and which one do you think is best for dendritic cell or antigen presenting cell targeting?
Well, we could do we could plan a seminar on this question with like 3 or 4 speakers. It's a complex question, so let me try to dissect. So you can give vaccine ID intramuscular or mucosal. IV vaccine, so far nobody's doing that, right? So let's start from the classical things. ID, it is generally more painful than IM. It's more reactogenic and is more difficult to administer. Unless there is a real improvement of the immune response ID, generally the IM immunization is favored. Most of the time, despite all the literature saying that ID may work better for dendritic cells, we don't see a big improvement on ID versus IM. I'm talking about all vaccines, not only self-amplifying RNA. If IM works, it's really the easiest way. Mucosal doesn't work very well unless it's a live attenuated. It's very difficult. We don't have mucosal adjuvants licensed, so it's a very difficult thing to touch. But in terms of systemic vaccination, IM is favored, not because it's more efficient, but it's because it's more practical and sometimes is less reactogenic. I think in Japan, they give IM some vaccines, and in the rest of the world are given, they give ID vaccines, and the rest of the world are given IM. I should check again that, but I think they were doing especially in the past. So it's not impossible to give ID, but generally IM is favored because of it's more practical and less reactogenic. For LNP-based RNA vaccines, there are 2 options at the moment that have been extensively tested. Most of it are given IM, and they're already reactogenic. So I suspect ID may be too reactogenic. And the other option is to give them IV, but for a different purpose. The vaccines are given ID, and there is also a phase 1 trial to induce the expression of proteins, eukaryotic proteins, not antigens, like monoclonal antibodies or proteins that have a role in the body. And why is given IV? Because there are some specific LNP formulation that are really not reactogenic at all, so they're not good, they're not used for vaccines, they're not immunostimulating at all, but they can be given IV. It is known that, I'm not talking about self-amplifying, I'm talking about mRNA in this case. It's expressed in the liver, and from the liver, you can have expression of proteins, including monoclonal antibodies. There is a vaccine trial from Moderna on chikungunya monoclonal antibody in phase 1. They could show that they could deliver the mRNA with a special LNP, a different LNP, IV, to express the protein in the liver. That I haven't seen, I don't think there is any trial in human that use IV for vaccination. I don't know if this will really help to have a better response. The self-amplifying RNA, I can tell you, you express your gene of interest, but you also express other proteins. So for all, imagine for all vaccines that express proteins that are not of human origin, being vaccine antigens or non-structural protein. If you give it IV and you express this in different tissue, different organs, you risk to induce a CD8 response against that. You have to be that. This has to be clear. So I don't like much the IV administration of RNA vaccine that express proteins that are immunogenic, because I think it may create a CD8 response systemic in the body. That's the risk here. When they do it with antibody, the mRNA only express antibodies, not very immunogenic. It may work. It may reduce the risk of a CD8 response against all the cells that express the RNA. It's my opinion, but we need to be checked. But I think that IM for vaccines, it's okay for data we have. You don't add much with the intradermal, and that will go IV only if you have to express something that does not induce an immune response, but is needed to play a role like an enzyme, a human enzyme or a monoclonal antibody. That's my opinion on that.
Thank you so much.
So thank you for this lecture, and that raised a lot of questions from the audience. I would like to thank also our YouTube viewers, and the YouTube live streaming is going to stop now.